So what I've done, I've set up just in case you didn't get everything when we went through the hard handout. I have it on the screen, but don't feel that that's something that if you got all the notes, you're all set. Hi, Hannah. I appreciate you joining.
Thank you, of course.
Well, you guys are the best. I honestly have to say, this is like one of the classes that I really look forward to every week.
Me too. I love this class.
Oh, I'm so glad to hear that. What I was saying earlier on is that I totally get it that you guys have a lot going on. So if at any point you have to leave or take a break, don't feel bad about it. I do record these sessions as well, always. And what I have on the screen, you should be able to see, is just a handout that we used when we went through the material in the class about the heart. So on the other side of that handout, I have a side about the blood vessels, and we'll just use that to guide us today. I'm so pleased to see you. You have no idea, I did actually miss this class yesterday. We'll just give a couple of moments for the last ones to join us, because I know that sometimes it takes a little bit of time, and when there's the most inconvenient time, the computers end up doing updates and all sorts of things like that.
Good.
So thank you again for everyone who has joined. I was saying that we'll just give a couple of moments for the very last folks to join us, because I know how it is when you think you've got everything set up, there's always an update or something. So I just want you to know that you have time if something happened to join us. I do always record these sessions as we've been doing throughout the semester. So if for some reason you were to have to leave early or join us only for a portion of the session, you should be able to still stay on track completely fine about what we're covering. What you should be seeing on the screen while we're waiting for the last folks to join is the completed copy of the handout that I gave to you when we talked about the heart. So on the reverse side of that, I have some of the key points that you might want to learn about blood vessels. So as always, we'll go through that. I think that we'll save going through the answers for the portion on the blood vessels till tomorrow when we meet at the class. So just so you know, you will get all the answers there. And if for any reason you miss those, they will be also on the recording as well at the very end of this session. But before we get too far, is there anything that I can help anyone with? Everyone's good, everyone's kind of staying on top of the workload. Any concerns, anything super exciting, happy that you want to share? Well, it sounds like everyone's doing good. I have to say that this is really my favorite class. So I'm super glad to see all of you. I missed you all on Tuesday. And I saw that 2 of you already had a chance to do the virtual lab. So there's pretty good lecture video or pretty good lab video that I prepared to go with it. So if you didn't get a chance to tackle that yet, and make sure that you find time to do that before end of this week. So that's what's going to make the attendance for the Tuesday session. What I'm going to do, I'm going to jump in to another view to share with you. And what I was hoping to do today was to go through mainly the lecture content on the chapter on the blood vessels. So on Tuesday, of course, we talked about the pulse. And if you didn't get a chance to do that, just remember that it is there I think that after that lab, you'll be pretty much experts on that field. So today we'll kind of just jump through that part real fast. We won't spend a huge amount of time on that. And tomorrow we're going to use as a chance to kind of tie all the loose ends, whether it's about this lecture review or about the Pulse lab. And also I have an activity where you actually get to really apply the information that you've learned about the blood vessels in the human body. So there's lots coming on and lots of good stuff. And as always, if you have any questions at any point, just jump in, unmute yourself, shout out. You can use any means to catch the attention. I really do want you to get a chance to catch me as soon as you have a question rather than waiting till the very end. Just a couple of reminders, and I'm sure we've talked about these already. The exam and practical results have been posted for quite a while, but make sure you take the time to look through those individual feedbacks. I really do try to take time and make sure that I provide individualized feedback, especially on the short answer questions. So do take time to look through that. And in terms of if you have missed any assignments, you feel that you would really do well if you could retake some of those, always remember you can just reach out to me and we'll work a plan how it works best for you. I've been talking, especially at the beginning of the semester, about the tutoring sessions or tutoring that we have available through the college. But remember that I'm here as well for if you need one-on-one or group time, if you have anything that you're kind of still feeling that, oh, you have further questions about what we've covered in this class. But really, what's probably the most important part is that what I'm asking you to do this week, it's going to be the quiz and practice anatomy. Those are the familiar parts. And the online lab is the thing that's going to be a little different. That's what we were doing yesterday instead of having a face-to-face class. So having said all that, what I'd like to do is talk through in the next hour or so about the key things you need to know about blood vessels. And again, a lot of the answers on that handout that I've given out is going to be from this review. I will make sure tomorrow that you get a chance to get answers to that if you didn't pick them during the talk, also from the handout that I'll show as a completed version. But it's always useful when you get to fill in things as you hear them, and kind of it gives you the skeleton that you can use to follow this session with. So, so far, we've talked about blood and we've talked about the heart, but we haven't really talked about the blood vessels. And that's the third and probably just as important component of the cardiovascular system. So if we made that analog that the blood was the transportation media and the heart was the pump, well, blood vessels are really the tubes within which all of this travels. So that's going to be the final part that we'll use to complete our system when we talk about cardiovascular system. And of course, there's a lot of clinical relevance for you. There can be many different kinds of bleedings. Bleeding sometimes if it's underneath the skin or so, bruising. We might have issues with high or low blood pressure and so on and so on. So there's a lot that really is directly relevant in clinical practice as we talk about this chapter. Before we get too far, what I wanted to do was to ask you if you could on the chat or just by shouting out, let me know about what type of blood vessels do you think that we have in the body? And I'm talking in terms of the general names of the blood vessels. You don't have to list any particular locations, but what vessels do we have? I see arteries. That's absolutely correct. Well done there. Do we have any others than arteries? Veins. Awesome. You're on a roll. There's something more than arteries and veins. There's something more that I'm still chasing after.
Capillaries.
Capillaries. Awesome. You guys are on a roll. You totally got this. So usually when I ask this question from my students, I get approximately these three answers: that we've got arteries, they're the blood vessels carrying the blood away from the heart. And oh, I love Caitlin, you're a couple of steps ahead than where I was already gonna go. So we have arteries carrying the blood away from the heart, and we have veins that carry the blood back towards the heart. And then we've got capillaries somewhere in between. And that's usually all I get. But we're going to do a little better on this class. We're also going to include arterioles and venules. And as we talk through those, it will make sense to you that why these are just as important part of the cardiovascular system and blood vessels as the ones that we immediately end up thinking. I also wanted to mention in terms of the what I have here on the slide, that most of the time, and by this I mean within the systemic circulation, arteries do end up carrying oxygenated blood, and veins end up carrying deoxygenated blood. But there is a special circulation in the human body where this is not the case. This is flipped upside down or in reverse. Does anyone remember we had our systemic circulation? We do have another circulatory circulation where we would actually end up finding it the reverse way around. Does it ring bells to anyone where we would see that arteries carry deoxygenated blood and veins oxygenated? How about the pulmonary circulation? Yes, that's where things get a little bit flipped other way around when we talk about what the blood contains and what type of blood we have. And then the really important part to keep in mind is that capillaries are really the business end, if you wish, in terms of where the exchange happens between the blood that's being transported within the blood vessels and then the cells or interstitial fluid that's around them. So that's where the largest amounts of exchange of materials takes place. One thing that I love about anatomy and physiology is that a lot of the time, even though we see all these separate little structures, we can find bigger rules of thumb, if you wish, that apply to most of them. And that's also true when it comes to the blood vessels. If we look at the blood vessels in general, pretty much all of them have 3 layers that we can find. And there's some exceptions to that. But we can kind of generate a general model of a blood vessel where we find 3 layers. And these layers are known as tunicas, and that's a term that I think comes from Latin and refers to this kind of a piece of clothing from the Mediterranean area, ancient piece of clothing that was very popular. So basically like a piece of fabric folded in half and head through the hole and arms on the side. So one way to think about those is that similar to having that layer of clothing, blood vessels are made of these multiple layers that are piled on top of each other. So if that helps you to think of that, use it. If it doesn't, well, you learned something about history. So the 3 layers that we end up finding when we describe the structure of the blood vessel, and we're going to start from the innermost part of the blood vessel. So from the lumen of the blood vessel, and move our way out from there, layer by layer. And the innermost layer within the blood vessels is known as tunica intima. And let's have a look at a few of the characteristics that make tunica intima very worthwhile for us to know. The way I think of that is that the term tunica intima refers to the fact that it's in intimate contact with the blood within the blood vessel. The other thing that's very worthwhile to notice in terms of tunica intima is the types of cells that this layer is made of. And if you look to the left upper corner of this slide, you'll notice that I've kind of sketched a thin layer of cells. Does anyone remember when you go back to the BIO181 or BIO156 or whatever introductory bio course you did, these kinds of cells, do they ring bells? Do you kind of, I see a little nodding going. So we talk about simple squamous epithelium when we talk about these, or endothelial cells. And the simple refer that we have single layer, and endothelial and the squamous cells refer to the fact that they're these kind of flattened discs if you look at their appearance. And this helps us to really understand what is the function of tunica intima. Well, first of all, tunica intima serves role in ensuring that the flow of blood within the vessel is smooth. So there's really no resistance to it much. It protects the layers that are going to be deeper within the blood vessel wall, and so on and so on. The other thing that this simple layer of flattened disc-shaped cells allows us to do is to make sure that materials can easily pass through this layer. So it's excellent for ensuring that we can exchange materials into the blood and out from the blood. So that's our tunica intima, the innermost layer of all the layers that we typically find within the blood vessel. If anyone has any questions about tunica intima, just shout out. Otherwise, what I'm going to do, I'm going to move on to the next one. And we're going to have a look of a layer called tunica media. So this is the middle layer, typically the thickest out of the three. And it's made of smooth muscle and some elastin as well. But the smooth muscle there is really the most important thing. And you remember that these smooth muscle cells, they were involuntary. You found those in the digestive tract, for example. And of course, now we're finding out that you found them around blood vessels. And the reason why we have this muscular layer around blood vessels is going to be that we want to be able to control the diameter of the blood vessel. So these smooth muscle cells within the tunica media allow us to do vasodilation and vasoconstriction. So basically, dilation make the blood vessel diameter grow, and constriction to narrow the diameter. So based on do we want blood to flow to a certain area or not, how fast we want it to flow, and so on, we're able to actually respond on a blood vessel level to the needs that the body might have, where the blood needs to go. So that's our tunica media. And the last one that I wanted to talk about is tunica externa. And I always get a little bit of kind of like, oh, how much extra should I throw to these slides? But I have gone ahead and added another name for tunica externa. I found that tunica adventitia is the same term that is a term that we use for the very same structure, and it's probably more commonly used in British-speaking part of the world, or English-speaking part of the world where British English is more common. So don't get thrown off if you see textbooks where either one, externa or adventitia, is being used. You can think of it being the same thing. And when we try to figure out, well, what does tunica externa do? It helps us to remember that a major part of that is made of connective tissue. And we talked earlier on at the very beginning of the course of different tissue types and how connective tissue really did the job of connecting things. We had special types of connective tissue, like we talked about the blood being a special type of connective tissue, but here connective tissue is really connective tissue as we think of it. So tunica externa plays role in reinforcing the walls of the blood vessel, but also anchoring these blood vessels to their side. We do end up finding nerves and lymphatic vessels also within this layer. One thing that kind of is a challenging question, but a question that I think you guys can figure out, is that, remember when we talked about the heart and how every single tissue in the body requires a way to somehow get nutrients, oxygen, get rid of the waste products, and so on. Well, if we have blood vessels that are very, very thick, we end up with a situation that that blood vessel might need blood supply itself. So that's where the term vaso-vasorum comes from, that we might have, especially on the largest blood vessels, this need to have blood supply to the blood vessel wall. Why do you think that it is mainly within the tunica externa or tunica adventitia where this is an issue? Why is this not an issue with tunica intima or tunica media? Feel free to shout out, or if you're like, that's a good question, then you can just be like, yeah, I agree. That is a great question. Well, remember that the blood flows inside the blood vessel. So tunica intima is gonna be directly in contact with the blood. And tunica media is only one cell layer away from the blood supply. But once we get further away to the tunica externa, now we're so far away from that blood, especially if we have a super thick tunica media layer, that we really need to come up with some sort of a solution, some sort of a way to ensure that the rest of the blood vessel gets blood supply as well. So this is why tunica externa is where we sometimes find these additional blood vessels to supply the blood vessel wall itself. I hope that that made sense. I know that it's kind of like we're starting to go from something very general to very specific examples, but that's anatomy and physiology. I'm going to show one slide and then we'll take a break for any questions. But when we look at this diagram where I have a picture of artery and a vein, one thing that's striking on this is that we find these same three layers, whether it is an artery or whether it is a vein. So really, I'm not making these up to confuse you or make your life miserable. Sometimes by making simplifications, it's easier to tackle all of the special adaptations. And this is where the special things come. You'll notice that capillaries instead have only one layer at their very thinnest, only one layer of cells around. So this is the time when I said that there's an exception to this rule of general structure of the blood vessels. Within capillaries, we typically end up finding only the tunica intima layer. Now at this point, I know it's been a lot of information. Does anyone have any questions about anything at all that we've covered? I see heads being like, we're doing good. If you have any questions, don't hesitate to shout out. What I wanted to do next is that we're going to go through all of the blood vessel types. And once we've gone through all of the blood vessel types, we're going to then jump to the physiology side of the things and have a look of how the blood vessels and physiology, things that go with the blood vessel physiology, what are the most important things about that. So the best place for us to start would be, of course, with the arteries. If you're ever asked, and I know that I don't do on this class that, but say if you go to the university and you're ever asked to discuss a blood flow, for example, through the heart or blood flow through the body, what's generally accepted or expected from you is that you follow the path of that flow. So if you just jump around and talk different parts, the grader might feel that, oh, I don't know if they really know how these go together. So always try to follow like a story arch, the topic, from the beginning to the end. So arteries were the first vessels directly after the heart. And it was within the systemic circulation where these are going to be carrying oxygen-rich blood. A couple of things that make arteries so special or so different in terms of their anatomy is their physiological needs. So first of all, we notice that this blood coming directly out of the heart has a high blood pressure. The other thing that we remember when we talked about the heart doesn't pump blood out at the constant stream. It's much more pulsatile how the blood comes out. So as a result of that, arteries must have adapted to that. So they are exposed to a high blood pressure and very pulsatile flow. Well, that brings us to ask that, well, what would then arteries have done to adjust to this? And that would be a great question, wouldn't it? Well, we have the answer here on the screen. I have a microscopic image of two vessels next to each other. So whoever acquired this picture either lucked out really lucky or just did a fantastic job finding an artery and vein next to each other. So now what I'm going to challenge you is to, and you have 50-50 chance, so you can't go wrong much. I'm going to challenge you to guess which one of these would be an artery and which one of these would be a vein. So you'll notice that the other one is pointed with blue arrows, other one with white, or the other one is on your screen on the left-hand side, other one is on the right-hand side. Which one do you think is the artery? Perfect. Thicker is artery. Good job. So we'll notice that the artery wall, arterial wall, it's actually physically thicker than the vein. The other thing that we notice is that this microscopic image that we have, blood has been drained, of course, out of these vessels, but the artery wall still maintains its shape, nice and round, even when there's nothing inside. Whereas the vein kind of tends to collapse. And this is what happens. I told you that I spent most of my years and funding my way through the med school and so on by doing postmortems and working with catheters. We actually see this with catheters that when you compare arteries and veins, arteries are typically nice and round in shape, but veins seem collapsed once we have drained all the blood out of them. This is where I kind of feel bad. And I feel that I can't wait for the COVID to be done so we can actually really do so much more. But I am grateful that we're able to do some dissections on this class. And once we are getting to a dissection where we get to compare arteries and veins, I really encourage you to feel it. Because anatomy especially is a field where just looking at diagrams is never going to be enough. You need to get the three-dimensional shape, but also simply by feeling the arteries and veins apart, you're able to tell the difference. So that's why I find it hard to think of anatomy as only a field that you could learn based on pictures. You really need to get into there and get to feel those. And that's what I did in all of my exams at the university. I was just like, well, can I feel it? And I'll tell you which one it is from the catheters. So there's three types of arteries that I want us to talk about. If you're okay with me moving ahead to that. Any questions? No? We seem good. Let's have a look of three different kinds of arteries that I want us to know a little bit about. And the first ones that I want to talk about are going to be our elastic arteries. These elastic arteries, an example of these would be, for example, our aorta and its many branches. So these are the vessels that are located right after the heart. So the very first arteries after the heart. They are characterized by having a very thick wall, the lumen, so the very center of the blood vessel, that facilitates a nice flow of the blood, so there's very little resistance. And one term that's sometimes being used when we describe these elastic arteries, we describe them as conducting arteries. So you might be like, well, what does that mean? Why do I care? Well, it's going to become more obvious once we get to the next blood vessel type. But what we'll notice here is that there is two big adaptations. First of all, there is no vasoconstriction, or the blood vessel, the elastic arteries, they themselves don't really systematically start controlling their diameter by that smooth muscle layer that we talked about. Rather than controlling the diameter themselves, they are very, very elastic, so they're able to expand and recoil as needed. So they serve as a way to kind of deal with this high pressure pulsatile flow, but also to respond to it and kind of maintain that pressure within the vascular system. So if there's a high pressure, they expand a little bit. If there's a low pressure, they're able to shrink a little bit in their diameter. So they're really doing a good job of conducting the blood onwards. They don't really do much of processing, choosing where the blood would go. They simply conduct the blood onwards. As a contrast to that, I wanted to talk a little bit about muscular arteries. So muscular arteries, these are most of the arteries that we study and we name. They have the thickest layer as the tunica media. So you remember this is where the smooth muscle was. And they have some little elastic tissue there as well. But the big thing there is that they're able to do vasoconstriction and vaso-dilation. So to control the diameter of the blood vessel because of this smooth muscle that we have within the muscular arteries, tunica media. Well, by doing that, by either kind of dilating or shrinking, they're able to control where does the blood get distributed. Do we direct a lot of blood to one part and less blood to another part? So that's the big difference between the elastic and muscular arteries. I hope that that makes sense. I see nodding. I'm very grateful. Thank you for immediate feedback. The last of the three that I wanted to talk about is, of course, going to be our arterioles. So eventually these end up being just a single layer, the tunica intima, when we get to the capillaries. But there's one special characteristic with arterioles that I want to speak about. And this characteristic that I want to speak about is going to be the pre-capillary sphincters. So you'll notice how where our arterioles are ready to turn into capillaries, we have these little muscle bands right where this junction would be. These are pre-capillary sphincters. So they're basically just muscle bands that can either squeeze and close that branch of the capillary network to be closed, or they can keep it open and allow blood to go to that part of the capillary network. What's interesting is that we actually have more capillaries in our body than we have blood for them. So you can never have all of the capillary beds open at one time. We need to choose where we put our blood to. So the choosing process is largely done by these pre-capillary sphincters. So that's why I think it's really, really important that we take the time on this review session to appreciate that that's what they do. And here you see nicely if they are closed, well, now blood simply flows through this region of the capillary network. We're not really letting the blood to wander off to where it's not needed. So there's this constant choosing where blood needs to go that's going on. Now that we've talked about arteries and arterioles, do we have any questions about anything at all that we've discussed so far? There's a lot of things I can help with questions in life, but I'm going to try to stick to the questions as it relates to the chapter content. I think we're doing good. Well, in that case, let's talk a little bit about our capillaries. And like I said, these are the kind of microscopic, very, very small vessels. The smallest of capillaries are only of a diameter of one red blood cell being able to squeeze through at the time. The other thing that we notice that the wall of a capillary is often just made of the tunica intima. So there's not really other layers there. This is the main site for the exchange of the materials. So these supply the cells and interstitial fluid immediately around them. And we find capillaries pretty much almost in all parts of the body, so pretty much everywhere. So based on everything that we've learned so far on this course and everything you know before, what sort of things could we want to exchange between capillaries and the tissues around them? Just shout out anything. Yes, absolutely, Stephanie. Well done. So gases, we want to make sure that tissues get oxygen. We want to pick up carbon dioxide. Remember, absolutely, Michaela, I'm so glad you said that because we've been speaking all about the hormones. Well, now we see that this is really why the hormones were circulating, because we want to get them to all parts where they might need to go. Of course, in addition to oxygen, carbon dioxide, hormones, we need nutrients, and we might need to get rid of all other waste products than just respiratory gases. So there's a lot of materials that we're exchanging here. So it's really the place where the passage of small solid and fluid particles takes place. And you're not going to be shocked when I tell you that I want to talk about 3 different kinds of capillaries. Why is it that it always comes in 3s? But it does. So let's have a look at 3 different kinds of capillaries, because not all capillaries are alike. And the first capillary type that I want to talk about is going to be called continuous capillaries. If you look at the walls of the continuous capillaries, it's very much of this unbroken layer. All of these cells are packed closely, tightly next to each other. So it really forms this kind of a sealed vessel. We find continuous capillaries typically, for example, in skin, muscles, lungs, central nervous system. I'm trying to think what else. Blood-brain barrier is a good example of that. Are you familiar with what blood-brain barrier is? Does it kind of ring bells, or you're just like, I don't know? Well, I'm going to just really quickly give a summary. So you remember if we talked about on the BIO201 about how brain had so much special needs in terms of there needed to be constant blood supply, not too high blood pressure, not too low. We needed to avoid anything that shouldn't be getting there, brain being very sensitive to certain things. So there's this special adaptation that actually separates that there's no direct flow as such to the brain. So there is this blood-brain barrier that kind of controls us. So this is where we would see the continuous capillaries also being present. Well, the next one, that was kind of a side tangent. Don't get too worried about that if that made no sense. The next one that I want to talk about is fenestrated capillaries. So again, you know that I'm a bit of a geek if we hear words and I'm like, where does that word come from? Because every single word in English is a new word for me. So I think in French, fenestration refers to a little window. So when you look at fenestrated capillaries, it's almost like it looks like a Swiss cheese. So it has all these little windows, if you wish, within the wall. So unlike continuous, that was just all solid cells there, we have now these little openings there. So places where we find fenestrated capillaries is places where filtration, absorption, secretion, things like that would be important. So for example, kidneys, intestines, places where hormones are secreted and so on. And the last one that I'm going to talk, we're going to up the game to even more than fenestrations. We're going to talk about sinusoidal capillaries. And you remember the term sinus. We've spoken about that in the class. Sinus was some kind of an empty space when we talk about many uses, many places where we can see that being used. So these sinusoidal capillaries have huge portions of the wall really missing. Places where we find sinusoidal capillaries would be places, for example, especially where we're clearing blood. Liver would be a good one. Bone marrow, spleen, and so on. So what happens if we have a blood vessel that's missing chunks from the wall? Well, the speed of flow through that blood vessel will slow down. Now the flow will be slower than it would be if we had a continuous capillary. Well, this works out actually quite nice, because now that this flow is slow, we have more time to really exchange materials and process that blood through these sinusoidal capillaries. I hope that that makes sense. The last one that I want to speak about would be the venules and veins. But do we have any questions at all before we move on to those? No. Okay. Let's have a look at the venules and veins. So veins, of course, we remember they were carrying the blood towards the heart in the systemic circulation, deoxygenated blood. One important thing to notice when we talk about the veins and we try to see the differences is that this is where the blood pressure is significantly lower. So you might be wondering, well, why does this matter? Why does the low blood pressure matter here? Well, when we look at the wall of a vein, what we'll notice is that these walls are much thinner than what we saw at the arteries. We talked about that when we compared those two microscopic images. If there was now a very high blood pressure, it'd be easy for us to burst those walls broken by accident. So actually, what we end up finding is that the blood pressure is low in the veins and the walls are thin. And it turns out that most of our blood vessels are actually, and most of our blood in the vessels is at the veins. So this is where I've got a question for you. And the question is, again, one of those. That's a really good question. I wish you would have told me the answer, but let's try. So now we've learned that the blood leaves the heart through the arteries. It travels everywhere in the body. We get to the capillaries. Now we need to get the blood back through venules, veins, and back to the heart. But how do we get the blood back to the heart? From, let's say, from your big toe. That's far away from the heart. You will have to work against the gravity. There's everything that could possibly make that job hard. Oh, I'm loving Caitlin. Good job. You know where I was heading with that. So what happens? There's a special adaptation for returning blood to the heart. Before I show you what it is, I feel so glad you knew it. Normally, my students don't know. They tell me that old blood pushes, the new blood pushes the old blood back to the heart, or the heart sucks the blood back in to itself. It's really, those play minor role. The biggest role for return of the blood to the heart is thanks to something known as venous return. So this concept of venous return is really, I think, one of the clever examples how body has been able to come around with a challenge. So what we end up finding is that the veins had thin walls, and a lot of the time veins get actually physically squashed by skeletal muscles or other muscles, so they get all the time a little bit squeezed. Every time that a vein gets squeezed, that blood gets squeezed upwards within there. And to prevent the backflow of the blood, we have something that we've seen before when we talked about the heart. We saw valves. Well, we see these valves also on veins. So they prevent the blood from returning backwards. That gets stopped thanks to these valves. And with every time that we squeeze these veins, a little bit more blood gets squeezed back towards the heart. And if you really think about that, well, I think it's easy to see with me because I get excited about things so much. But if you think about that, we rarely are completely still. We constantly tend to move a little bit. If nothing else, when we breathe, we actually end up squeezing some of those important veins that help us to carry the blood towards back to the heart. Same as when you're sleeping. I used to work in a sleep lab. You never sleep totally still. You're constantly moving a little bit. So all of that little moving helps us to pump the blood back to the heart. And there's two clinical things that I want to highlight of this. So I've shared with you that I used to live in UK for quite a while. We love military parades and stuff like that. And every once in a while you see someone standing there and they go down. Well, what happens with this individual is that if you stand, especially your knees locked, and you're really trying not to move, you're not doing any of that venous return, and your brain is then not getting adequate amount of the blood from the heart because heart is struggling to get enough blood to pump. And we always want to preserve brain before everything else. So the easiest way for the body to deal with that is that let's make you lie flat real fast so that the blood can get to the brain. So that's what we do. That's why you pass out. It's a really quick way of dealing with that issue. So the other clinical relevance to the venous return is, of course, varicose veins. And these varicose veins are consequence when we have these valves, but some of them fail and the blood ends up pooling lower and lower to wherever the valves are working. So you end up with these poolings of the blood within the veins. And there's many factors that play a role why some people get varicose veins, some don't. Even as far as I think that professions have been sometimes suggested. I have a lot of friends working in aviation, and apparently it's more common to get varicose veins if you're in high altitude for a big portion of your life. I don't know enough about that, but that's what I've been told. So that's really everything that I could think of in terms of the anatomy. If I really want to summarize what you need to know, does anyone have any questions about any of that? Well, because I'm feeling a little low, a little lonely, I thought that we should do at least an icebreaker. What is your least favorite food? Feel free to throw it onto the chat or just to tell with a voice. What's your least favorite food?
Fried pickles.
Oh, pickles, that's a good one. Any others? I have one that's I really don't know how to deal with. Sauerkraut. Has anyone of you ever had that? That's tough. Well, that had nothing to do with what we were doing. I was just feeling lonely. So let's have a look of a little bit of the physiology side of the things. So a couple of terms, couple of measurements that I wanted to talk about. First of all, in terms of the blood flow. When we talk about blood flow, we're talking of the volume of blood through a structure in a given time. So typically it's milliliters per minute, but there can be other measures. But what I really want us to focus on is the blood pressure. And what blood pressure really measures? It measures the force that the blood pushes against the wall of that blood vessel at a certain spot. And usually that's measured in millimeters of mercury. So that's where the mmHg comes from. What we're seeing when we're measuring blood pressure, we typically get two values. You get a higher value and a lower value. And this is the time when I'm going to be like, oh, I wish that you would be able to help me because I'm really confused. Why do I have those two values? I'm not confused, but I want to know if you know it. Does anyone know it at this point? It doesn't matter if you don't know. Why would we have two values for blood pressure? Well, remember when we talked about the pulse when we did the lab, the virtual lab. And on that lab video that I prepared for you, I had these two graphs that I drew. I was so proud of myself for taking the time to draw them. So you remember when the ventricles were dilated, they were not squeezing, the arteries, they're going to be nice and just their normal shape. Then the ventricles really contract. The arteries might need to expand a little bit beyond what they would normally be to facilitate that high amount of blood that's coming there real fast, real strong. So when we're taking the blood pressure, where we're measuring this force put against the blood vessel wall, well, we can't really take it on one and not the other one. So that's why we measure it on both of these. So that's why we're ending up with the systolic value, which is the higher value during the ventricles squeezing, and then the diastolic value when the heart relaxes and ventricles refill. So we get two values. And there's many factors that affect the blood pressure. But what I wanted to ask from you guys is that sometimes I go to a doctor, and I've spent a big chunk of my life surrounded by doctors, surrounded by nurses, but I go to see a doctor, and all of a sudden my blood pressure is much higher than it would be at home. Does anyone know what that would be called? It doesn't have to be scientific. Well, one term that we use sometimes to describe that is white coat syndrome. So really, you can get high blood pressure just by stressing out of things. Like I think I've told you that, you know, when you get reflexes tested, you're worried, is my knee gonna jerk the right way? And you can really hype yourself. You can really hype up your blood pressure as well. So if you feel that, oh, my blood pressure is through the roof, take a deep breath. Start with that. The next thing that I would normally speak would be all about the pulse, which really tells us we can use it to find out, for example, heart rate and so on. Since we did the entire lab on that, I'm just gonna, at the end, link a video to that lab if you haven't already had a chance to look at lab video. And what I want to talk instead is the way we can regulate blood pressure. And we're going to start by looking how we regulate blood pressure in short term. And this is the perfect time for me to say, I told you so. Remember when I told you that the process of homeostasis and homeostatic feedback mechanism loop, that will haunt you till the very end. Well, this is the time when it comes up again. So just a really crash course to recap. Homeostasis was all about maintaining consistent internal environment in the body. And the most simple way to explain homeostasis was this diagram that we did together and we figured out. So first of all, there's some sort of a change that happens. So body is always interested about changes, not so much about constant things. If something changes, that perks our interest. We detect it in one way or another. So there's some sort of a sensor or receptor or detector. That message gets processed in the control center. And you remember that was typically our brain. Within the control center, we have a set point, something that it normally should be. You could think of it like when we talked about thermoregulation, the AC control unit on the wall. And if it's off, the stimulus is off from the set point, we act on it. And most of the time, the way we acted on that was by stopping that from happening. So negative feedback. There were 3 times in life when we saw positive feedback. Does anyone remember that? What those 3 were? I see nodding. You remember, good. But most of them, yeah, lactation, perfect. Yeah, childbirth, perfect. And blood clotting. So really, all other times we are trying to stop that change from happening. Well, life gets easy now that you know this homeostatic feedback mechanism, because we can use that to explain the short-term regulation of blood pressure as well. So I was all about making your life easy by making you learn this first. So we have a change in blood pressure, and we're comparing that change to what would be the set point. I think it should be 120/80, but I haven't updated that slide in a while. But we have something that we would think that is the normal blood pressure. And there's some sort of a value, some sort of a change, and we want to make sure if it's no longer what the set point is, well, that's when we are going to get busy. So we somehow notice this change, and we have various different kinds of receptors, and some of the receptors that are especially relevant here are carotid receptors at the carotid sinuses. So you remember when your common carotid artery coming up at the neck divided into external and internal carotid arteries, there's this division point, and there we see this little expansion within there. If you do dissection, you actually see there's a little bulging there, and that's where our carotid sinus is located. So it's kind of a dilated area where the common carotid divides into two. We also have receptors for regulating blood pressure at the aortic arch. So you remember aortic arch had all these three major branches, and within there we have receptors that regulate also the blood pressure. And the final place that I wanted to mention is, of course, our large vessels. They have receptors for blood pressure. So now we're somehow detecting that there is some sort of a change, and we pass that message to our control center, which is the medulla oblongata, so the medulla part of the brain. That's where we have the regulatory function for the blood pressure. And if we end up noticing that the blood pressure that we've noticed or detected is not matching within our set point, and we are saying that this is way off, we're going to act on it somehow. Some of the ways that I wanted us to discuss how we can act on blood pressure changes include prescribing medication, or what would probably make more sense would be to treat the cause of the high blood pressure or low blood pressure. Of course, in short term, your blood vessels themselves can do vasodilation or vasoconstriction as a response as well. So rather than spending pages upon pages of talking of this, you'll see how convenient this way of summarizing everything with homeostatic mechanism loop diagram came. Well, at least I still believe in it. I choose to believe in that. There is a long-term regulatory mechanism for the blood pressure. This works largely through the kidneys. So we change the blood volume, which then would change the blood pressure. And we do have two mechanisms there that we can use. We have a direct mechanism, which is all to do with eliminating the volume, through, for example, through urinating, or preserving some of the volume by kind of conserving the water. And then we have an indirect mechanism, which is much more complex. That's known typically as renin-angiotensin-aldosterone mechanism. Real mouthful. There is a full lecture video where we actually look into that. But at this level, when we're doing review, I'll just leave it to that. And we just acknowledge that it does exist. But what I really want to talk about or highlight as kind of the big take-home message of the blood pressure is that we want to have a high enough blood pressure so that we are really able to perfuse the tissues in an adequate amount. But we don't want it to be so high that the blood vessels would get damaged. So the goal is really to find that golden balance between the two. And does that always happen? Well, it does not, unfortunately. Again, I was worried that I'm going to be lonely today, so I had icebreakers. But I'm not going to ask you to tell, but I was like, well, what's your favorite smell? But we can talk about that on the class tomorrow. Think of your favorite smell and we'll talk about that. What I do want to talk about is all the many things that can affect your blood pressure. So there's a lot of things that play a role in there. But what I think is going to be most important for us is to talk about when the blood pressure is either too high or too low. So let's start with elevated blood pressure. I think that Typically, the clinical criteria for that has been 140/90. That changes a little bit depending on who you ask and what year you ask. They do tend to change those numbers around a little bit, but it is something that would be higher than a normal blood pressure. So what we end up noticing is that this is dangerous for many, many reasons. It can lead into heart failure, vascular diseases, renal failure, so kidneys fail, they filter the blood, or a stroke. What happens is that the heart actually ends up having the heart muscle growing thicker. So your heart is working harder, it's thicker, but it's also weak, even though it's thicker, the muscle there. It's really a flabby, if you look how it functions. So when I used to make my money and my salary by looking at people's hearts, just because you have a big heart, it doesn't mean that you're super healthy. It can mean the exact opposite. Your heart is just working really hard because the blood pressure is so high. So you end up with this thickening of the walls of the ventricles. Not great news. What could you do, or what can it cause? There's risk factors for that. It can be hereditary. So again, you can blame your parents for everything in life. But in addition, diet that's high in fat and cholesterol, obesity, age to a certain extent. Also, diabetes has been suggested to be a predisposing condition, stress, smoking. But most oftentimes, there's no clear cause for hypertension. It's just something that you have, and we can't really pinpoint that this is why you have it. But what you can do, you can eat diet that's lower in salt, fat, cholesterol. You can exercise, lose weight, stop smoking, take medications if needed, and so on. So that's hypertension. We're going to talk about hypotension in just a second. But before we do, I'm going to ask you, which one would you pick? Hypertension or hypotension? Too high blood pressure or too low? If you have to choose one.
High blood pressure.
Oh, don't pick high blood pressure.
No,
That is
maybe low.
Pick low. Because
No.
I love you were like, I'm gonna go for it.
Seemed fun for some reason.
I agree. You're like, I want to be extra rather than just being like low. Well, it turns out that hypotension, which is low blood pressure that's like below 90/60 or something in that scale, for the longest of time, we were like, what is the danger with that? That we are going to find out a danger. But we still cannot say that for most people it would be an issue unless your blood supply is so low to the tissues don't get enough blood. Of course, then it's going to be a problem. But actually, it seems, based on some of the newest research, and this is not for fact, this is just research. We need a lot more research to be certain, but it seems that having a low blood pressure can be actually beneficial. It can be associated with an extended lifetime expectancy. People with lower blood pressure have less cardiac illnesses. So if you have to pick one over another, pick that. There's 3 different subtypes of low blood pressure, and I know that we're approaching the end, so I'm going to be real fast. So the reason for low blood pressure might be that it's just temporary. So you, for example, get dizzy when you stand up. Things like that. There can be chronic low blood pressure. Usually this is due to having poor nutrition, or it can be associated with risks of developing many other serious conditions. And then we have something that's known as an acute hypotension. So this indicates that you are about to go into a shock. And the reason why I wanted to talk about this is that I want you to know the signs and symptoms of the shock. So we love making fun of police constables in UK. So every mnemonic that we have is about police constables. So PC-FATS is our way of remembering signs and symptoms of a shock. When you're about to go to the shock, or your patient is, their skin is going to look very pale. That's because there's very little blood flow to their skin. So it's not this nice and reddish. It's really whitish. The skin will also feel cold and clammy. Again, there's not a lot of blood coming in there, so there's no temperature or temperature being carried there, heat being carried there. Kind of sweaty, clammy. The breathing is going to be really fast and shallow. So these individuals, although they breathe fast, the breathing is not very good. It's just very, very shallow, like dogs. And of course, the pulse, when you measure that, it's going to be fast, but it's going to be weak. It might be really hard to find it. You might actually really have to work to find the pulse of an individual who's about to go to the shock. The other thing, if you think that someone's going into shock, how are they going to feel about you coming and testing them, all of that? They're not going to be loving it. They're going to be agitated. And they're also often thirsty. So they're wanting to get something to drink. These individuals are at the risk of going to the shock. And if we were in a class, I would show you exactly how to treat someone who's going into the shock. But we'll see if we get to that tomorrow. Otherwise, I'll tell it to you later. What I do want to share with you real fast is that we control the blood flow constantly. So there's constant regulation of that. And we have two mechanisms for that. Extrinsic control of the blood flow is all about using your sympathetic nervous system and hormones to control that, whereas the intrinsic regulation of the blood flow is through kind of capillary control at the local level. So meeting the local needs. And what we're going to be doing tomorrow, we're going to be looking at circulation at various different special parts of the body. So for example, skeletal muscles, normally about 20% of the blood is dedicated to skeletal muscles, but it can go up to 70%. So we'll look at that tomorrow. With the brain, we discussed that there needs to be a constant blood flow there. It needs to be steady, not too high, not too low. Neurons die real easy if there's issues with the blood flow there. Skin is excellent for thermoregulation, so getting rid of extra heat or saving the heat. Also acts as an amazing blood reservoir. So a lot of the blood can be stored within the skin. Lungs needed to have low blood pressure, because if we had high blood pressure, we broke those little blood vessels there and you're going to drown onto your own blood. That's not a great way to go. And of course, we talked about the coronary circulation to the heart muscle itself earlier on. The only thing that I want to really emphasize at this point is a question that I love asking in an exam, is that when we speak about the speed of the blood, what we end up finding that speed of the blood and blood flow in the vessels, it's fastest right after the aorta. That kind of makes sense. Aorta, that's where in the arteries, we're pumping the blood out, it's moving the fastest it can. That's the fastest place. The slowest speed of the blood is within the capillaries. And again, that should make sense, since we remember that capillaries were the site where the exchange. See, I'm a guy, I have to stop to do writing. I can't talk and write at the same time. That's where the exchange of materials takes place. So we really want the flow of the blood to be as slow as possible so we can do as much. And I still couldn't write it correctly. That was tough for me. But we have to have a slow speed so that materials can be changed in capillaries. One of the great questions to ask is that, well, why is the flow of the blood so low in the capillaries? It comes down to the fact that speed depends on the cross-sectional area of the blood vessels. So you notice that we have most of our blood vessels are actually capillaries. So because there's so much capillaries, the blood flows slower through them than through others, where we have less of them, so they can actually pick up the speed again. What I'm not going to speak about next to you is about hydrostatic and osmotic pressures. We'll leave it for when we talk about the lymphatic system. So the forces that push blood into the blood vessels and out of the blood vessels, only thing that I'm going to share here is that sometimes some of the components of the blood leak out of the blood vessels. And this is usually the components that would be same as your blood plasma. So how you can tell that someone has an edema? That's because there's interstitial fluid. Plasma has leaked out of their blood vessels, rather than that they're just chubby. Well, what do you do if you push that area and you release? That area will retain this indentation for a while, even when you release that. So that's one of the signs. Usually also that it's not symmetrical on both sides and so on. So really, if you learned nothing else from this chapter, these are the key points that I really want you to remember. Arteries typically run deeper because we don't want to cut an artery. If you cut an artery, you're going to bleed and a lot and real fast. Veins, sometimes they are deeper, but a lot of them are also superficial. Remember, there's low blood pressure, so we don't really want to be squeezing those veins really hard. Veins also, when I teach my students arteries and veins, and we actually study their anatomy, where we find them, vasculature in the body, I tell them, learn the arteries, veins, you can always wing it. That's not really being a good teacher. That's kind of being a very horrible. But because veins have so much more variation, and we learn things in a classroom that applies to an average human being, but you'll end up finding that veins are not always there where you would think they are when you go to clinical practice. Arteries are there where you think they are. So learn the arteries first, because they're much more likely to be where you're looking for them to be. And then remember that the brain has this unique drainage system. So we drain the blood out of the brain in a very unique manner that's different from other parts of the body. Same goes for the intestines. So when we are grabbing all those nutrients, energy from the food that you're eating from your digestive tract, pretty much all of that blood goes first to liver. And the blood goes to liver because your body is greedy. If you give it energy, it will just have cells, let's go crazy and burn off that energy. What we want to do, we want to direct that blood to the liver, who's the only sensible organ in this system, and stores the energy until you need it later. So that really summarizes everything that I had on mind to tell to you. And you'll be glad to know I don't have any more icebreakers. This is it. I know that we almost made it to that hour and 15 minutes, but I'm sure that no one's going to be super upset if we wrap up a little earlier. I so appreciate that you joined me today, especially knowing that my boss was here. So I promised I have a treat for you for tomorrow, because I believe in bribery and blackmailing. And also, we do actually have a really fun class lined up. We will take the time, if you didn't get a chance to do the lab virtually, you can do it in the class. And I also want to make sure that any questions that you have, including going through to the answers to this handout, we'll go through them in the class. So I'm not going to keep you any longer because I know everyone has things to do. I really, really do appreciate that you showed up today. You guys are amazing. I'm not lying when I say that this is like my favorite class. This is the class that I wish that I could only do. So thank you for joining me tonight. Thank you for being so good on this class. You really are all doing fantastic. I'm super proud to have you in this class. And if you have questions, I will be sticking around. But if you don't have, this is it for me for tonight. I hope you have a good night. Thank you.
Thank you.
Thank you.
Thank you so much. Thanks. Thank you. I so appreciate you. And that really completes our recording for tonight. I appreciate all of you who either joined me in person or through a recording. And I really look forward to seeing you tomorrow. Have a good rest of the night.